Bismillahirrahmanirrahim. Today we learn about the male internal genitalia. So at first come to the testis. The testis is a male gonad, and in female the homologous part is the known as ovary. And the testis is a mobile organ and it lies in each half of the scrotal sac, and it is suspended by a spermatic cord. The main function of the testis include the production of the spermatozoa and secretion of a hormone that is known as testosterone, for which the male pattern is seen. That means the testosterone is responsible for the development of the secondary sexual characteristics of the male. Now come to the size and measurement. It is almost oval or ellipsoid in shape. The length is 5 cm, breadth 2.5 cm, thickness 3 cm, and weight is 10.5 to 14 cm. What are the parts? It has two ends, upper end and lower end, two border, anterior and posterior border, and two surfaces, medial surface and lateral surface, and one epididymis. Let's see the testis. So this is the testis. So we told the parts of the testis. It has two pole, upper pole. It has a lower pole or lower end. It has two border, anteriorly anterior border and posteriorly posterior border, and it has two surface. This is the lateral surface and the opposite one will be the medial surface, and one epididymis. This yellow color structure is known as the epididymis. This is the epididymis. Now come to the description of this. So upper end. The upper end is overlapped by the head of the epididymis, and the upper end gives attachment to a fibrofatty body known as appendix of the testis, which represents the embryonic remnant of the cephalic part of the Mullerian duct. And the spermatic cord is attached to the upper pole of the testis posteriorly. The lower end, it is connected to the tail of the epididymis, and the anterior border, it is smooth and convex, but the posterior border is broad and flat and awkward posterolaterally by the body of the epididymis and posterolaterally by the vas deferens. The medial surface is smooth and convex, and the lateral surface, it is overlapped posteriorly by the body of the epididymis, and which is separated from the testis by a semilunar recess, which is known as the sinus of the epididymis. That means the testis, it is separated posteriorly from the epididymis by a semilunar recess, which is known as sinus of the epididymis. And this sinus of the epididymis, it helps to determination of the side of the testis. That means it is a right-sided or left-sided testis. That means sinus of the epididymis will always lies posterolaterally. It lies posterolaterally. Now let's see, so it will be more clear. So we are talking about the parts of the testis. So look at that. The upper pole, it is attached to the head of the epididymis. The lower pole, it is attached to the tail of the epididymis, and the anterior border is free, smooth and convex, and posterior border is related to the body of the epididymis. And we told that the testis and the epididymis is separated by a recess, which is known as sinus of the epididymis. So this is the sinus of the epididymis. This is the sinus of the epididymis. And one thing we told that the sinus of the epididymis, it helps to determination of the side of the testis. So sinus of the epididymis, usually it lies posterolaterally. So let's see. So we told you, let's see how to determine the side of a testis. So I told you that the anterior border is free. It will be present anteriorly, and the posterior border is attached to the epididymis. So as this is attached to the epididymis, this will be posterior border and this will be the anterior border. And we told you that the sinus of the epididymis, lies posterolaterally. So we can see here there is the sinus of the epididymis. So definitely it will be the lateral surface and other surface will be the medial surface. Why it will be the lateral surface? Because we already told you that sinus of the epididymis lies posterolaterally and posteriorly it is attached with the epididymis. So definitely this will be the lateral surface. Now let's know about the epididymis. What is epididymis? It is a comma-shaped body and it is situated along the lateral part of the posterior border of the testis. And it consists of three parts: head, body and the tail. Now what is the function of the epididymis? Function of the epididymis is storage and maturation of the spermatozoa, absorption of some fluids and addition of some substance to the seminal fluid to which there is spermatozoa is maturating. Now the head, we already saw that the head is connected to the upper pole of the testis and it is attached to the upper pole of the testis by efferent ductules. And sometimes the head of the epididymis contains a body which is known as appendix of the epididymis. And the body and tail, they are made up of a single highly coiled duct of the epididymis from which the vas deferens emerges. Let's see. So we are talking about the epididymis. So look at that. This is the head of the epididymis, this is the body of the epididymis and this is the tail of the epididymis. And look at that. The head of the epididymis, it is attached with the upper pole of the testis by some ductules. These ductules are known as the efferent ductules. And then in the body and tail, these ductules are continued as a single duct. And from the tail, this duct is continues as a name of the ductus deferens or vas deferens. And sometimes in the head of the epididymis, it contains a fibrofatty mass or body which is known as the appendix of the epididymis, which is not present here. Now we are talking about the testis. Now let's learn what are the coverings of the testis. The testis has three coverings from superficial to deep. These are the visceral layer of the tunica vaginalis, tunica albuginea and tunica vasculosa. So visceral layer of the tunica vaginalis, it represents the lower persistent portion of the processus vaginalis. Processus vaginalis, it is a part of the embryological remnant, and this tunica vaginalis, it covers the whole this visceral layer of the tunica vaginalis, it covers the whole testis except in the posterior border, because in the posterior border there is attachment of the epididymis. Now come to the tunica albuginea. The tunica albuginea is a dense white fibrous coat which covers the testis all around, and posteriorly this tunica albuginea is thickened and form an incomplete vertical septum which is known as the mediastinum testis. And from the mediastinum testis, numerous fibrous septa arises and it goes to the inner surface of the testis and ultimately divide the testis into 200 to 300 lobules. Later we will see it. And the innermost layer is the tunica vasculosa. It is a vascular membrane which lines the individual lobules of the testis. So At first, see the coverings of the testis. So the coverings, the first outermost layer is the visceral layer of the tunica vaginalis, then tunica albuginea and tunica vasculosa. So this is the first layer. Then the tunica albuginea, this is the tunica albuginea, and tunica vasculosa, these are the some vascular membranes. Now look at that. The tunica albuginea, which is a white fibrous mass which is thickened posteriorly and forms a vertical septum which is known as mediastinum testis. And from the mediastinum testis, numerous fibrous septa arises and divides the inner surface of the testis into multiple lobules, about 200 to 300 lobules. So these are the lobules. If you see, this is the mediastinum testis, and look at that. From the mediastinum testis, numerous fibrous septa arising and divide the testis into 200 to 300 lobules in each testis. So this is a single lobule. This is another lobule. Now come to the structure, histological structure of the testis. At first we will see the macroscopic structure. That the macroscopic structure, that means each lobule, how does it look like? Each lobule of testis, it contains number 1 to 3 seminiferous tubules, number 2 the interstitial cells of the lady. The seminiferous tubule, it consists of some convoluted portion in front and some straight portion behind. This straight part of the seminiferous tubule ascends in the mediastinum and join with the adjacent seminiferous tubule to form a plexiform of network which is known as the rete testis. These in turn, that means the rete testis in turn give rise to 12 to 30 efferent ductules which emerges from the upper pole of the testis and enters into the head of the epididymis. That means through these efferent ductules, the upper pole of the testis is attached to